Cancer is a result of genetic changes to DNA. As the understanding of genetic changes improves, the number of targeted therapies are rapidly growing. Thus, the therapy can be tailor-made for the individual depending on the genetic profiling. Testing to acquire molecular profile data has become essential to determine the best personalized therapy for your patient. To expedite the collection of critical therapy-relevant molecular data, oncologists are turning to Gopro Focus Fusion Target Test based on next-generation sequencing technology. Gopro Focus Fusion Targeted Test can deliver a whole panel of biomarkers in one go rather than looking at one biomarker at a time. The panel testing approach is strongly recommended by relevant treatment guideline approved by the FDA. Today, there are already genes target associated with NSCLC targeted therapies, while many others are under investigation in clinical trials. But the current method of running the individual test for each gene has two significant drawbacks. First, it can take weeks to sequentially separate tests, possibly causing a dilemma for you whether to wait for the complete biomarker result or save time and choose a potentially suboptimal therapy. Second, the amount of tissues required are often not available, potentially requiring the patient to undergo a painful rebiopsy and delaying treatment decision. The Gopro Focus Fusion Assay Test helps remove these barriers by using next-generation sequencing to simultaneously deliver multiple biomarkers results for multiple targeted therapies from a single sample. The Ion Torrent NGS technology, using a chip containing millions of where separate sequencing reaction occurs in parallel, allowing testing of multiple genes and biomarkers at the same time. This is important because most NSCLC tissue samples from biopsies are very small and don't have much tumor cell content, and these small samples can't be analyzed by every NGS test. The Gopro Focus Fusion test requires only a small amount of tumor's DNA or RNA isolated from the tumor cells and therefore can successfully analyze those small samples, allowing access to appropriate therapy for more patients.